So this session is going to be the first session, Advanced Neurophysiology Devices and High Throughput Acquisitions. So we'll have 3 talks in this session. The first talk is going to be by Lili Karashchuk, who is here at Allen Institute for Neurodynamics. And the talk is going to be about motor observatory capturing diverse mouse behavior. This is in collaboration with Sam Sober. And we'll hear from Lili, please.
Thanks for that introduction. Can you hear me? Yeah?
Yes.
Okay, cool. One problem that I'm interested in terms of neuroscience and measuring behavior is really this sensory motor loop. I think it's crucial for understanding motor movements generally. So beyond what's happening in the brain, I'm really interested in what's happening in this whole loop from the spinal cord to the muscles to the world, to the mechanosensors and proprioceptors. And it's a really exciting time. This loop is crucial, I think, for all kinds of motor disorders and for understanding how the brain controls movements in general. And I think it's a really exciting time for investigating the sensory motor loop. Here we are at this open data in neurophysiology, and there's so many new models coming out. So there's new biomechanical models of the rodent body, as well as, I don't have shown here, but as well as the fly body, and some things coming out for all kinds of animals, horses, zebrafish. There's myomatrix arrays for recording motor units, which I'll talk about a bit later, and new tools emerging for markerless 3D tracking to really characterize the space of behavior. And to me, this is exciting because so far, I want to show this graph. So this is a graph of BMI versus abdominal circumference. And we know those two are related. But if you looked at, perhaps maybe you might control your study for a specific range, you would find a very low correlation. And to me, this really illustrates how much we need the full space of behavior if we want to understand motor control. And so here we are. So in the past, I'll go for a couple of different mouse studies. I don't know what happened, but we'll just go with it. So here we have a few different mouse studies from the last decade. A couple have focused on reaching, and they focused on using marker-based tracking, and they could track 1 or 2 points on the mouse and could track the whole reach. The one at the bottom, the video that played here at the top, they tracked the whole mouse movements, walking, it was just forward, and 1 dot for each paw. More recently, we have dance, which has come out. There have been new tools for markerless tracking, and here you can see you can actually track the whole mouse body, but still it's limited to this open field arena. So we are moving forward in terms of getting more and more interesting, more complex behaviors. But I think we could push it even further. So that's what we set out to do here. So we've been building a motor observatory, and the idea is to have this multi-camera rig and have as many different objects as possible and set up this mouse playground. It's transparent, but you can see the mice moving in all kinds of different objects and have the whole, as many different kinematics as possible. And so the structure of this talk, I'm going to talk a bit. I will talk about the mouse, the motor observatory. Then I'll talk about some improvements we're making to 3D motion tracking to make it possible and to make future studies like this even easier. And finally, on behalf of Sam Sober, I'll talk about the myomatrix arrays, which we are setting up in this motor observatory as well. So let's start with the motor observatory. So I'm going to go through the process of designing it and the toys within it. So here we had an initial playground. It looks something like this. I essentially got stuff from BioServe and put them in. But I found there was a couple of issues. I mean, the mice interacted with it fine, they love it. But you can't really see when the mouse goes underneath, you lose track of it from the top cameras. Or if the mouse goes on top of the treadmill, you lose track of it. And then if you want to record with cables, essentially none of these will work because you have these holes. So I started to design a couple of different objects. So we had cylinders, some kind of running wheel, and I'll go through each one in the rig, the latest one. So here's the small triangle on the left. You can see the mouse going up on it and down. And we have cameras both from the top and on the bottom, so you can track the pause. We're tracking, well, I'll talk about the tracking in a moment. Here you see the big triangle, and the mice love this big triangle in the wheel. Here we have a small wall. The mouse climbs that. Yeah. It's really fun to see them balance on it. And they do this without any, I mean, they're intrinsically motivated to explore. They learn. There is a bit of a learning component with exploring some of these, but I mean, they're just ready to go. Here's a big wall. So we had an early version with a lot of holes, but we realized that it was hard to see through it, so it limited the range of vision for the rest of the rig. So we've expanded to that. I don't know why that paused. You can see here the mouse climb. And it takes them a session or two to learn this, to climb up, and then another session or two to learn how to climb down. And so you'll see the mouse on the right hasn't fully learned how to climb down yet. So it's gonna fall, yeah. But yeah, I think it's an exciting avenue for exploring movements. And here finally is a running wheel. This one I got off the shelf. It's pretty standard. But yeah, they love to run on this. Finally, we've been messing around with a treat dispenser. We're still figuring out how to make this work well. But right now we found that the mice like to just spin it, which is a fine motor task. Yeah, and they eat like one pellet maybe the whole session. Yeah. So here we are. And so then the question is, how do we analyze the movement? And I'll talk a bit about the 3D tracking pipeline we have, and then how we go through it and the results. So the pipeline is, as I developed, Karashchuk et al. 2021. It's called AnyPose. A bunch of different labs are using it now. The idea is you have some calibration videos. So you have a Charuka board that you can print out, or here for flies, we had to custom order it. And then you record your videos of your animal as well. You pass these through Deep Lab Cut or some other 2D tracker like Sleep or Lightning Pose. You have some filters, and then using the calibration and the refined detections, you triangulate, and then you can optimize this triangulation, with an optimization, and then you can compute angles based on inverse kinematics. So all of that together, we started to track the mouse. So this is the key points that I settled on. I think essentially I tried to get every single joint that I could see. So it's a lot on the digits here, 3 on the tail. Here's a couple of different ones. This took a lot of annotation. Okay, and then you can see some examples of tracking. So it works really well in the open field, as you can see here. And here's some in the initial playground as well. I think it's exciting. You can actually track through the objects. The mouse walks around. There's a couple more. And then you can see here we're still having some issues, and I think this showcases the limitation of the current motor observatory if you want to build something like this. One issue that we have is we don't have enough views right now pointed at these objects, so we're adding more views. We have 7 cameras currently. We're adding more from the sides. Another issue, I think we just need to annotate more, and yeah, we've been doing that. So, I mean, this brings me to the improvements to 3D motion tracking. So right now I've been using any pose. I think you can get similar results from dance, honestly. But really, we need improved annotation in 3D. I think I really would like to reduce the amount of annotation that has to be done. I'm sure maybe some of you have done behavior here. Who has annotated frames of animals? Yeah, that's quite a bit. So you know how time-consuming it is, and how many frames you have to annotate beyond what you imagine. So I think this is really huge. And then finally, we've been working on improving the behavior visualization and annotation of the behavior downstream as well. So I'll show some examples of that. So here's the app. So I've been building an annotation app for 3D annotation, and it's fully web-based. You just go, it's nice, you don't have to install anything. And then you can zoom in, as you can see, and then you just go and annotate. So this has helped us annotate a lot of frames very effectively. It's available right now, it's got some documentation. And the cool thing here is, so that was just one view, right? But as soon as you start to annotate multiple views, it will give you feedback on whether it's consistent across cameras. So you see here I've messed up, and I've annotated the left ear and the right ear. I've switched them. That's a common mistake. And so I will correct them now. And now the points are green. So this is a really useful sanity check, and I've imported some previous data that I've annotated without this, and I've noticed, oh, I've made some issues. It's usually like for flies, I would switch which leg. Like it's a little confusing which leg you're annotating sometimes. In mice, often the left and right is confusing. And sometimes you don't have the point, it's a little ambiguous, like that spine point is a little ambiguous, like where to place it, and having it consistent across the views, I think, gives you more consistent tracking. In the end, it's really about the annotation. Okay, but even so, it still takes a lot of annotation. I mean, even if you have a great tool for annotation, it's still going to take some time. And this is across all the tools. So if you look at deep lab cuts, these is from the papers themselves. It takes about 800 frames to get to human variability, maybe 1,000 frames here in dance. And these are 1,000 multi-view frames, so you have to label within each view. It really balloons quite quickly. Similar with lightning pose, maybe it cuts it down by half, but you still need to label frames every time you have a new setup, every time you have new animals, new rigs, and it really is time consuming. And so what we saw, I've been visiting these computer vision conference, CVPR, and trying to understand what's new, what can we use. And I'm really excited about this new tool that's come out. It's called CoTracker. And the idea is that you annotate just the points in the first frame, and it can propagate those tracks through the whole video. And if this could work well, then potentially we could really dramatically reduce the amount of annotation we have to do. We could just annotate the first frame in each video. And then maybe we could even use this for tracking. So this is a project that I've been working with Katie Rupp here at the Allen, software engineer. And the idea is something like this. So the current pose estimation takes in an image and predicts a pose. And then again, the next image completely independently predicts a new pose. Every tool works like this right now. Sleep, Deep Lab Cut, even Lightning Pose. Lightning Pose has some pose-to-pose like filters. I mean, any pose also has some filters, but fundamentally the deep network is like this. Point tracking like CoTracker, I should just show you the demo. It takes a first image and then a pose on the first image, and then a new image, and you can predict the pose on the next image. And the idea that we're working on, and I won't have time to go into too much detail here, but feel free to ask me more about it, is we're trying to integrate these two approaches so that you can have an image and then it predicts on the first image, but it can also propagate based on the image and the predicted pose. So integrating point tracking and pose estimation together into a more robust approach. But there's still some limitations, right? So here, I run CodeTracker on the mouse data, and so far CodeTracker is trained on a bunch of natural kind of images and doesn't quite generalize to this experimental setups. You see some of the points drift. And so what we've been working on with Jonathan Wong, another software engineer here, is we've been trying to model the setups across, we're trying to get a lot of different data sets, multi-view data sets of animals, and modeling them so that we can generate one data set for point tracking across all these experimental sets, and then we can have like one model to do point tracking that works really well for our data. And so here we can actually model the rat in 3D with Gaussian splatting, and the idea is that from these videos we can propagate these points through time, and then we can use that to train the network. All right, and then finally, once you have the tracking, so let's step back, you have to annotate the behavior as well. And so here's another tool. This is part of any pose already. I still need to do a better job documenting it. That's a crucial part of open data. But it's already available. You can visualize multiple views very easily. You can jump to points. You can zoom in. So if you have a long video, you can zoom into a specific point and then annotate the ethogram. This is crucial for, I think, this kind of naturalistic data that is becoming more and more popular. All right, and in the last couple minutes, I'm going to talk about the myomatrix arrays that are emerging as this new tool for measuring motor signals. What we've seen so far is there's been a rapid progress in neural recording methods, but EMG has lagged behind. It's still stuck for a while on this groups activity of like motor unit spikes. So you can't resolve individual motor unit spikes from this implanted fiber. What we really need is this high-resolution EMG that can work during active movement across species and behaviors. And that's what the myomatrix arrays provide. Whoa. Okay. So the myomatrix arrays consist of this ground pad, and then I think of them, it's a little bit, I think of them as kind of neuropixels for muscles. It's less contacts right now, but you can go up to, so it's, here's an example array with 4 threads, and they have 8 electrodes, 8 contacts per thread. And using these, you can actually distinguish individual motor units. I don't know. Okay. But yeah, you can see here the motor. So here's a fine wire electrode, and you see the grouped activity of the motor units. But with the myomatrix arrays, because they're so close together and so small, you can actually distinguish the spikes of individual motor units that are causing the muscle to activate. So I'll just present from this very quickly. Okay, that works. So it's been tested across many different species. So here's a mouse triceps, which we're also testing out at the Allen. People have done it on the mouse masseter, the rats, triceps, the digits, the pulpus spongus muscle in the mouse, and the cat soleus muscle, and then on songbirds, vocal and expiratory. I think Sam is very interested in this, and even in hawk moth larvae and frogs, and these work across all of these different species. And then I wanted to highlight a next generation. So this is the myomatrix arrays are already 2 years old, and there's been some development, and they're coming out with the optomyomatrix arrays. I think they're very exciting. Essentially, it's just like the myomatrix arrays, but you have an LED as well, so you can implant it and potentially stimulate muscles or proprioceptors. or anything in the greater body. And here's just an example of us testing out the myomatrix arrays. I don't have the data from this yet, but you can see the mice are moving freely and it's implanted in their muscles. Okay, and I want to close check out the myomatrix booth. It's in the atrium. Turn to the right when entering the atrium. Daniel will be there, and we'll have all the different myomatrix arrays for you to see, and you can ask for more questions about them as well during breakfast, lunch, during breaks. Here you can see all the myomatrix arrays. And then I'll just summarize the Camber ones. So it's arrays across many muscles, different species. They've now supplied devices to 200 labs worldwide. All the devices have been free of charge to date, although they will begin selling them per the grant rules later this year. All right, and yeah, I'd like to thank the Neurobiology of Action team here at the Allen Institute for all the collaborations in setting up the Motor Observatory, as well as the Greater Allen Institute and people at Emory University, especially Sam, Gordon, Olivia. Yes.
Yeah, hi. So I just had a comment and then a question. Just the comment is that you, at times you had blocked images because of refraction of the light. I was wondering if you could index match by coating the plastic so that you get a better transmission. It's not perfect, but the other thing I was curious, because you brought up EMG wires, Sam Sobel's device, and also you have this very large set of tracking. Is there a data set in which you both tracked and measured the EMG and have some idea of the sensitivity that you get from tracking as opposed to the onset of muscle movement? Some metric of resolution?
Of the EMG or of the tracking?
Of the tracking. I mean, how strong a motor act do you need before you could actually see a change in an elbow joint move or the spine flex?
Yeah, I don't know. I don't think I've seen such a data set yet. I mean, we're clearly trying to collect it here at the Allen. I think it will become available in the next 1 to 2 years. I think just freely moving one will be available in the next year. I know Sam Sober is also collecting some data on like a mouse on a treadmill with the myomatrix arrays and 3D tracking. But yeah, I mean, it's all kind of emerging. We don't have that data set yet, sorry.
That's great. Krista? That's would that be possible?
Yeah, that's interesting. So you have multiple mice. I think it might work. You have to really set up a lot of things. So you see the Motor Observatory, everything is transparent. So they would have to really live in this transparent box. I don't know if they'd be okay with that, but maybe they would. And the other thing is like occlusions. So if you have multiple mice and they all close together, you might have some trouble tracking them. But in principle, it's possible. And I mean, the 3D tracking is coming now. You can do multi-animal 3D tracking. I would really just be worried about occlusions. Yeah.
1 minute, actually. I have a question myself. So when you showed us this sort of like a device that myomatrix, so it's wired, right? Is there any way to get it to like, wireless? Because that would be ideal. Then you can have this sort of like complex environment where you can track by video, and as David was saying, then you can actually link the actual movement with the muscle precursor of like, when this is happening.
Yes. Yeah, I would actually love it for it to be wireless. I don't think it's a requirement to link the behavior and the activity. I mean, we're doing it wired already. I think there's already so many objects you can have, even with wires, as you saw with the triangles, the wheels, and so on. But yeah, there's clearly like all the tunnels, we can't do that. It's coming. I think I've talked to Sam multiple times about this, and I mean, he's aware. But it's the problem really is the connector on the Maya matrix arrays. So it has to, I think they have to adapt it, and there's like a whole engineering process they make to build them. So I don't know when that will come, but maybe in the next year, if we keep pushing, we could have wireless ones, yeah.
Thanks, Lily. That was, I think this work is really exciting. Um, Coming from more of a preclinical or drug discovery aspect, there's always, with multiple cameras, the data load is quite high. Like in our lab, we do in days, weeks long, maybe even the entirety of an animal's lifelong recording. I'm wondering, like, where do you balance the need for multiple cameras, high resolution tracking versus just the practicality of collecting long-term data, and especially like compute costs? Do you think like these methods could be done on the edge, for instance, to really scale up the approaches?
Yeah, it's an interesting question. I feel like you might have a better answer to this than me, honestly. So in our setup, everything is compressed online. So we're able to collect 7 cameras from one GPU, compressed everything. So the data storage cost isn't too bad. But yeah, I mean, the processing cost is still quite intense right now. I think you could do it on the edge. But yeah, I mean, at the end of the day, like, I think we still need more efficient models for the post-tracking. I feel like that's a big bottleneck. Like, there are some on the edge for like 2D, but for 3D, it's still like, I think maybe the best is like 10 or 20 frames that you can process per second, but we're recording at 200, so it's like 10 times slower than what it is. Yeah, I really would like to push for that, but yeah, I don't know, it's either that or working on annotation. If somebody else is working on efficient post-estimation models, please let me know.